In Module 2, we focus on strategy, measurement, execution, and system design. We examine how organizations translate plans into actions and how performance management systems support this journey. Section 1. Meaning of performance measurement and management. Performance measurement refers to the process of tracking indicators such as sales growth, customer satisfaction, or operational efficiency. Performance management goes beyond measurement by using this information to guide actions, improve systems, and achieve strategic objectives. Section 2. Understanding effective strategy. Strategy is the long-term direction of a firm. An effective strategy is clear in purpose, adaptive to change, inclusive of measurable goals, executable with existing resources. Without execution, even the best strategy remains a plan on paper. Section 3. Methodology for executing strategy. Execution requires translating strategy into operational goals, allocating resources effectively, aligning people and incentives, monitoring progress with dashboards and scorecards, making timely adjustments. Execution is where many organizations fail. Turning plans into consistent results is the ultimate challenge. Section 4. Benefits of a strategy-focused organization. A strategy-focused organization, SFO, aligns everyone's daily tasks to the strategy, uses transparent measurement systems, encourages cross-functional collaboration, continuously learns and adapts. The benefit is that employees see how their roles matter to the big picture. Section 5. Building a comprehensive performance management system. A robust system includes performance contracts, agreements between managers and employees, dashboards, visual tools to track real-time performance, monitoring mechanisms, reviews, audits, and continuous feedback. This makes performance management systematic and measurable. Section 6. Managing organizational performance and strategy. Managing performance means regularly reviewing objectives, providing coaching and adjustments, celebrating achievements. Managing strategy means staying alert to environmental changes, revising plans when necessary, ensuring strategic consistency over time. Together, they create a cycle of growth and adaptability. Module 2 Summary. In this module, we studied performance measurement and management, effective strategy, execution methodologies, and the benefits of building strategy-focused organizations and performance systems. Course conclusion. This course has shown that strategy and firm performance are inseparable. Performance management is the bridge between strategic intent and organizational results. Module 1 highlighted the purpose, cultural alignment, behaviors, and tools like the balanced scorecard. Module 2 emphasized measurement, execution, and building robust systems to manage both performance and strategy. Together, these concepts prepare leaders to build organizations that are strategically aligned, performance-driven, and growth-oriented. White heavy checkmark word count, 5,600. Script 2, DNA typing, proteomics and beyond, recombinant DNA technology. Module 1, DNA typing and proteomics. Introduction. Welcome to this course on DNA typing, proteomics and beyond, recombinant DNA technology. In Module 1, we focus on DNA typing, proteomics, and their applications. These technologies form the foundation of modern biotechnology, medicine, and forensic science. Section 1, DNA typing, meaning and applications. DNA typing, also known as DNA profiling, is the process of identifying individuals based on their unique genetic makeup. Applications include forensic investigations, paternity testing, identifying disaster victims, tracing ancestry and population genetics. DNA typing relies on analyzing regions of DNA that vary widely between individuals. Section 2, Methods of DNA typing. Techniques include restriction fragment length polymorphism, RFLP, Polymerase chain reaction, PCR-based methods. Short tandem repeat, STR, analysis. Single nucleotide polymorphism, SNP, profiling. These methods vary in resolution, speed, and applicability, but all rely on detecting genetic variation. Section 3, Proteomics, exploring the protein world. While genomics studies genes, proteomics studies proteins, their structures, functions, and interactions. Proteomics is important because proteins carry out most cellular functions. Protein expression patterns reveal disease states. Protein networks help us understand systems biology. Section 4, Techniques in Proteomics. Key techniques include two-dimensional gel electrophoresis for separating proteins, mass spectrometry for identifying protein structures, protein microarrays for studying interactions, bioinformatics tools for mapping networks. These tools have revolutionized medicine, drug discovery, and personalized healthcare. Section 5, Applications beyond proteomics. Proteomics extends into cancer research, identifying biomarkers, pharmacology, designing targeted drugs, agriculture, developing stress-resistant crops. Thus, proteomics bridges genetic information and functional biology. In Module 1, we covered DNA typing methods, proteomics, and applications beyond genomics. These technologies allow us to identify individuals, study proteins, and design medical innovations. Module 2, Recombinant DNA Technology and Future Directions. Introduction. Module 2 explores recombinant DNA technology, the methods used to modify genetic material for research, medicine, and industry. Section 1, Fundamentals of Recombinant DNA Technology. Recombinant DNA technology involves cutting DNA with restriction enzymes, inserting fragments into vectors such as plasmids, introducing vectors into host cells, expressing new genetic material. This process allows scientists to engineer genes and organisms. Section 2, Tools of Recombinant DNA Technology. Key tools include restriction enzymes, molecular scissors, DNA ligase, joins DNA fragments, vectors, plasmids, bacteriophages, viral carriers, host systems, bacteria, yeast, mammalian cells. Section 3, Applications in Medicine. Recombinant DNA has transformed medicine by enabling production of insulin, growth hormones, and clotting factors, development of gene therapies, creation of DNA vaccines. It represents a leap from treatment to cure in many genetic conditions. Section 4, Applications in Agriculture and Industry. In agriculture, genetically modified crops resistant to pests and diseases. Crops with improved nutritional value. In industry, production of enzymes for detergents and food processing. Bioremediation using genetically engineered microbes. Section 5, Ethical and Safety Considerations. While promising, recombinant DNA raises ethical debates. Concerns about genetically modified organisms. Potential environmental impacts. Human genetic modification ethics. Regulation and transparency are crucial for public trust. Section 6, Future Directions, Beyond Recombinant DNA. Emerging technologies include CRISPR-Cas9 gene editing, synthetic biology, personalized genomics for tailored medicine. These innovations extend the legacy of recombinant DNA into new frontiers. In this module, we examine recombinant DNA technology, its tools, applications, ethics, and future directions.